5 minutes or so. Welcome, everyone. Some people have come to this special interest group before, and the first 5 minutes is kind of a repeat, but some haven't. So I'd like to do this at the beginning. So CASP has played a critical role in advancing the field of protein structure modeling and prediction, and in the last CASP we had an exploration of modeling not just single states of proteins but conformational ensembles, and we think this is a really important area of structural bioinformatics, but also of biology. So we've organized this CASP special interest group on ensembles of conformations from the perspective that proteins and nucleic acids, for that matter, are not single structures as we typically view them in their crystalline environment where most of the structure determinations have been done, but proteins actually sample a conformational energy landscape based on energetics and temperature and Boltzmann. And the protein is popping back and forth between some low energy states and even sampling higher energy states that often are very important to the way that the protein works. So this is really the structure of the protein. So we put together this group, and it's kind of redundant what I just said, recognizing that biomolecules don't have static structures, but rather adopt a distribution of conformational states. The CASP Special Interest Group is trying to approach how we can assess methods for predicting conformational ensembles or multiple conformational states, and even whether this is feasible. I have to say it's a little risky because it really is a challenge to go from the models to validation when you have conformational distributions, and we'll be talking more about that. So we'll look at developments in the field, both experimental methods and computational methods, and particularly methods from artificial intelligence and machine learning. And I kind of anticipate that this cycle of CASP is intermediate, and it really may be in the future CASP that will really make an impact with this program. So we have an organizing committee who are listed here. Egzi Karaka in Turkey, and Damiano Piovesan, Silvio, Emily, Wim Rankin, and Shoshana Wodak. And we kind of brainstorm what things we should present and how we should evolve it. And we'll be putting together the CASP competition or the CASP experiment. But we're very much guided by the CASP organizers, John Molt, Christoph Fidelis, and Andrei Kristofovich, who are very much involved in this. And if there's other people who really want to get involved in this, you could write to this email, which isn't my email. It's an email we set up particularly for this project. I say that these videos will be posted on YouTube, so you can get the links or the email address from there when it's posted. So what do we mean by multiple conformational states? Well, it might be a kind of basic idea like adenylate kinase, which fold flips between two conformations with the lid open and closed, so a two-state ensemble, to multi-protein ensembles, to even intrinsically disordered proteins, which would then sample wide ranges of conformational space. And that's actually a field of its own, and it really isn't the original focus of this program, but we are going to be looking at some of those kind of targets as well. It makes sense. I mentioned there's a YouTube site. You can just Google for it to search it, or take a screenshot if you like of this. With all the videos that we've done so far, there's about, I don't know, about 10. We've been doing it for about 10 months. And some of them are purely computational, like AlphaFold sample from Bjorn Wollner as an example. And some of them are much more experimental, like Susan Sudakawa talking about how you use SACs to characterize multiple conformational states of proteins. So these are available online. And in addition to our special interest group listed at the top here on alternate conformations, I just want to advertise that there are two other special interest groups ongoing. One is specifically about AI methods in modeling broadly. So it actually includes some of the same topics we cover, but it's much more broad: protein complexes, protein structure prediction, maybe even protein function prediction using AI methods. And Arnie Ellefson organizes that. And then we also have a special interest group on modeling and structure prediction of RNA, which is organized by Rachel Kretsch at Stanford. And these are the links to that. The CASP meeting, CASP 16, is, I don't know that we've cracked the hotel lined up quite yet, but I'm pretty sure it's close, if not done. So the dates are November 29th to December 3rd. And at that point, we'll have a session about how this experiment proceeded over the year. And I just mentioned that we record it. We'll get permission from the presenters to post the recording on YouTube. If you put chat comments in, we'll track those chat comments and make those available to people. And we also have a LinkedIn group, which I organized just to share papers and announcements and things like that. And you should feel free to post or join that LinkedIn group. Okay, so that's kind of the administrative background. So the schedule for today, the agenda is, I'm pleased to introduce Bowen Jing from MIT. Bowen is in the groups of Bonnie Berger and Tommi Jaakkola at the MIT Computer Science and Artificial Intelligence Laboratory. And Bowen and Bonnie and Tommi have put out a preprint recently, which the organizers thought was very exciting in terms of a new method to look at conformational ensembles. And then after the presentation, we'll have some time for questions. And then I want to talk a little bit about where we're at in organizing experimental data for the CASP 16 experiment. So yeah, so I'm going to let Bowen, if you take the control of the screen. AlphaFold meets flow matching for generating protein ensembles. And I think you should be able to share, Bowen.
All right. Thanks very much for the introduction and the invitation to speak at the seminar. So over the past couple of months, with my advisors, Bonnie Berger and Tommi Jaakkola, we've been looking into a way of combining the very well-established AlphaFold architecture with a technique from generative modeling of images, from the more consumer and enterprise-oriented AI community, in order to have a generative model of protein structures given a single sequence as a means of maybe trying to have a better performing or a more principled way of generating protein conformational ensembles while still using the very well validated AlphaFold architecture. So for this audience, I will not spend too long on the introductory slide. But yeah, so we're essentially starting from a point of looking at the kind of the current ways of using AlphaFold to obtain multiple structures for a single protein, in this case, the canonical adenylyl kinase, right? And these methods leverage the fact that AlphaFold is not using just a single sequence, but the entire MSA. And so by changing the MSA input, we can get multiple outputs. And these methods, which include subsampling, mutagenesis, and clustering, they work actually quite well for many cases. And there have certainly been very exciting publications showing cases in which this works. But there are some limitations to these methods that suggest they're not, the end of the story. And in particular, for computational reasons, there's also been the rise of protein language modeling based structure prediction architectures such as ESM-fold, in which case you do actually only have a single sequence, and you're not able to kind of induce stochasticity by subsampling or ablating the MSA. And additionally, maybe kind of more interestingly in the long term, AlphaFold at the end of the day has been trained on these crystal structures or these cryo-EM structures. And so the training objective is not to model the perhaps different conformational ensembles that you might think about obtaining, like for example from molecular dynamics simulations. Maybe at physiological temperatures there are certain transitions that you would just never see deposited in the PDB, And so no matter how much you change AlphaFold at inference time, it's unlikely that it will be able to give you that structure. So what we would like to investigate is to say, well, AlphaFold is being trained as a predictive model. A single input maps deterministically to a single output. And so far, our methods for obtaining a stochastic output from AlphaFold have all relied on changing that input, right? So we inject stochasticity by injecting it into the input. Is there an alternative way where we obtain stochasticity by making that part of the modeling process itself, right? Something that takes a single input, but then gives us a stochastic distribution of outputs. And that describes, in a nutshell, generative modeling as opposed to predictive modeling, right? So what we would like to investigate, and what the field has been kind of moving towards a little bit in the past year or so, is generative models for protein structure prediction. And just to be very concrete here, the problem definition has an input, which is a deterministic protein sequence and perhaps its associated multiple sequence alignment. But now we want the model to be able to give us an output which contains a distribution of protein structures rather than a single one. And there have been some works in this area, largely in the machine learning literature, that have tried but maybe not succeeded beyond some minimal threshold, because the modeling architectures that are used here are not AlphaFold, right? And so these models generally have struggled to even recapitulate the single structure accuracy of AlphaFold. And so, if the single structure accuracy is very poor, it's not really that meaningful or interesting to ask what is the distribution of structures that this model gives, because that probably more just reflects the model uncertainty rather than any kind of true structural heterogeneity. So the approach that we're going to take instead is to actually leverage AlphaFold itself, not change anything about the architecture, but just repurpose it as a generative model. And the way that this is going to be done is via a fine-tuning procedure that wraps AlphaFold inside of a training framework from the generative modeling literature. And that framework is, as the title suggests, flow matching. But here I'm going to just talk a little bit as a detour about diffusion, because flow matching is, in some sense, very similar and just a slight generalization of diffusion. And diffusion is more familiar. So that's generally the way I proceed with the exposition. So diffusion actually provides a very interesting framework where you can take any predictive model and easily convert it into a generative model without needing to change the architecture too much. And so that's kind of what we want, right? Because we have AlphaFold, which is a very, very good predictive model of protein structure. And if there's some way that we can turn it generative, then it would be possible to perhaps obtain higher quality structural ensembles than the previous methods in the generative modeling literature which have kind of tried to design their bespoke model architectures, which are not AlphaFold and not as well validated. And so how does this conversion take place? Well, to take a look at that, we kind of look at maybe the more common context for using diffusion generative models, which is for generating images, right? And this is a graphic that is typically used to illustrate what happens in diffusion. You start with a natural image, you corrupt it by adding noise, and then the neural network is supposed to remove this noise, right? And concretely, what is the neural network doing? Like, what are the inputs and the outputs of the neural network that's supposed to remove the noise? Well, it is taking as input a noisy image, and it's trying to predict as output the clean image, right? And it's doing so maybe with information about some text prompt. So at the end of the day, the neural network itself, the architecture itself, is something that outputs an image, right? Along with taking as input a noisy image. So that means that if you had some hypothetical model architecture which took some text prompt and tried to produce a single output image, right, a deterministic output image, then you could easily convert that into something that produces a distribution of output images by additionally providing as input to this neural network a noisy image, right? So this noise input is now providing the role of injecting some stochasticity into the modeling architecture, and hopefully that stochasticity is reflected in the output in a way that is consistent with the text prompt. And it's the role of the training framework to ensure that that is the case, right? That the right kind of stochasticity in the input is carried over to the output. But from the neural network architecture perspective, this is the only modification that you need to make in order to make a deterministic predictive model stochastic. And so this is in the image generation case. In the protein structure prediction case, we can apply a very similar recipe, right? So we have actually it's, yeah, this slide's probably better. So, right now we have AlphaFold, which is a deterministic model that takes in a multiple sequence alignment and optionally a template. And then it gives us, a deterministic output structure conditioned on those particular inputs. And this almost already looks like the recipe for a diffusion generative model. And all we have to do is to swap out this template input for a noisy input, where the stochasticity is, something that can be explicitly modeled at training time, as opposed to, having some implicit distribution over template structures. And as long as we fine-tune this in the right way, this will provide us with a sequence to structure diffusion model, or maybe more precisely, a MSA to structure diffusion model, right? So for a given MSA, the noisy inputs provide the role of injecting the stochasticity into the output structures without ever needing to change or resample the MSA. So this can be done for AlphaFold. It can also be done for ESNFold, where we currently actually lack even a template input, right? So for a given sequence, and we also lack an MSA input. So for a given sequence, there is literally only one possible output structure ever, right? So with the recipe, though, we can additionally provide this noisy input, and the stochasticity will be carried over to the output and hopefully gives us a reasonable structural ensemble. So to go into a little bit more detail how this process happens, I'm going to describe the flow matching procedure. First, the training of it, and then we'll look at the inference. So the general idea of flow matching, as the name suggests, is to learn some kind of a flow in this very high-dimensional space that pushes a noisy prior distribution, such as a Gaussian, to your data distribution. And the way that this flow is going to be learned is as follows. At training time, you start out by sampling random pairs from the prior and the data, right? So you sample a noisy image, and then you have a data point that you sample from your data set. You interpolate them to get some kind of intermediate noised image. And then you pass this into your neural network and try to have it predict that clean image, right? So it's a denoising model. Now, there are many paths that interpolate through this single noisy image, right? It's not possible to deterministically reconstruct the clean image from that partially noised image. So the model is going to learn something like an expectation of the clean image conditioned on the noisy image. And as you do this over the course of training and do this for all possible times and all possible noisy images, you will eventually have a neural network that implicitly represents this vector field. At inference time, you can follow this vector field to generate data. So you start out by sampling something completely noisy, such as the Gaussian, right? And then from that Gaussian, you predict the average image. Now, at this point, the average image is not going to be very interesting. It's just going to be completely gray, right? So not literally this image. But you sample the average image, and you take a small step towards that by interpolating. And then you reevaluate the direction of the vector field. And now the average denoised image will be a slightly more informative. And you take a little step towards that, and so on. And as you proceed through time, you will arrive at the data distribution on the right-hand side, assuming that your neural network model has been trained well. So this is the case for images. For the case of proteins, it's actually very similar, right? Except now the denoising model is AlphaFold itself, or ESNFold, as the case may be. So instead of a Gaussian now, we still define a Gaussian prior, but not a isotropic Gaussian, but a correlated Gaussian whose samples from that random variable look like kind of these random polymer coil structures, right? So they roughly capture the intuition that adjacent atoms in the amino acid sequence should be relatively next to one another. So we want that piece of information to already be there, but otherwise it's pretty much a very random and certainly not very realistic protein structure, very noisy. So then we take that, feed it into AlphaFold, or once it's in this flow matching framework, that's the method that we call AlphaFlow, right? Now the model will predict some kind of a mean structure. And so as you can imagine, in the case of adenylate cyclase or adenylate kinase, it'll actually predict the lid kind of halfway between open and closed, although that's not the structure that's shown here, right? But it'll predict that mean image or that mean protein. And then you interpolate towards that, right? Just like in the image case, you interpolate towards that. And then you interpolate, after you've interpolated a little bit, you reevaluate. And, based on the initial sample that you drew, this partially interpolated structure will look a little bit more similar to either the closed lid conformation or the open lid conformation. And that'll affect what the average protein structure predicted from this intermediate point is, right? So maybe in this case, the initial noisy structure looked a little bit more like the closed lid conformation. And so the second prediction will have that lid close a little bit. And then you keep on interpolating, and then you repredict, and so on. Now, at a very high level, the procedures for images and proteins are very similar in this manner. There are some subtleties, which I don't think I should take the time to discuss, given the time constraints. But I did want to briefly touch on what those subtleties mean for the training procedure, right? Because unlike images, we have a notion of equivariance or invariance when we're modeling protein structures. AlphaFold, for example, was trained with an invariant loss function. And so when we make our predictions, the predictions are kind of oriented arbitrarily. So how does that affect how we do the training and how we do the interpolation? So just want to talk about that very briefly. Namely, the way this ends up working out is that when we sample things during training, we have to do this RMSD alignment, right? We have to take the noise that we sample and align it to the structure that we draw from the training data before we do the interpolation, right? And then when we predict our output, well, because AlphaFold does not know any information about the initial orientation, the output is going to be randomly oriented. And so we still need to train with a invariant loss, like the frame aligned point error loss that is used by default in AlphaFold. And then at inference time, there also needs to be a RMSD alignment step, which is, after we predict our denoised image, we need to actually, our denoised protein structure, we actually need to reorient it so that it's RMSD aligned with the initial noise before we do the interpolation, and so on, right? Because the second prediction will still also be randomly oriented, and we need to just align that. Okay, so that was just a very brief overview of the method. I wanted to talk about the two kinds of evaluations that we do. And I think these relate to the very well to the slide that was shown at the beginning, where we have different time scales of these conformational transitions. So in the first set of evaluations, we're just going to be looking at, for proteins, for uniprot IDs that have multiple structures deposited in the PDB after the AlphaFold training cutoff date. What is the ensemble of structures that we predict for those uniprot IDs? And does that have good diversity or coverage of the structures that were deposited? And so you might think of these as being, certainly the longest time scale transitions. And then later on, we'll look at actually being able to train on molecular dynamics ensembles, where because of the limited time scale of those simulations, the conformational transitions that one would capture by training on those simulations is certainly limited in time scale, but maybe also informative in a different sort of manner. Now, for the PDB ensembles, I wanted to just bring up that the training procedure proceeds very similarly to the way that AlphaFold was pre-trained initially, right? We have a cutoff date, which is May 1, 2018 for AlphaFold, and we just continue fine-tuning the AlphaFold weights on the PDB, except now instead of doing the training procedure used initially for AlphaFold, we fine-tune it under this flow matching procedure, right? So all the other training practices remain the same. The way we're going to evaluate on these test set of 100 uniprot IDs deposited after the training cutoff is we're going to generate 50 structures and then compare it to the PDB ensemble of 2 to 30 structures by the precision, recall, and diversity. So the precision is, for each generated structure, how close is the nearest true structure, right? And so this may be the most the closest to the notion of single structure accuracy. Recall is the converse metric, right? For the structures that do exist, how close is the nearest generated structure? And then finally, diversity, as the name suggests, is just how diverse is the ensemble. And here we take 1 minus the LVDT so that higher is always better for each of these metrics. So this is the baseline, which is MSA subsampling, right? We see on the left that there's this very intuitive trade-off between precision and diversity. On the right-hand side, we might also expect to see a trade-off between precision and recall, as we saw in the transporters and receptors benchmark from Diego Del Almo's paper. However, the interesting thing on this particular subset, or rather this test set, is we actually don't see that trade-off. And it turns out that reducing the depth of the MSA doesn't actually increase the recall that much beyond the original AlphaFold. There's a little bit of an improvement, but there's actually also general deterioration. And that might be just a feature of the difficulty of this particular test set. Which also, to be clear, is not a cherry-picked test set. This was essentially I took all of the UniProt IDs with multiple structural clusters that were all deposited after that cutoff date, and then I took all the proteins that were under a certain sequence length. So with that being said, perhaps MSA subsampling doesn't seem to be giving us what we want on the recall, but at least we can see if the diversity comparison, right? Do we get a better trade-off between precision and diversity by switching from the MSA sampling to AlphaFlow? And we do see that that is the case, right? So for a given level of diversity, we're able to maintain much better precision than just reducing the depth of the MSA. On the recall side, the performance is kind of pretty similar to MSA subsampling. So I don't think there's too much I don't think there's really any meaningful conclusion to be drawn there. So clearly, getting a recall of these deposit structures is still very hard, even though the model is currently able to propose multiple hypotheses for these different structures in a way that maybe preserves the fidelity a bit more than MSA subsampling. We see similar results for ESM fold, but for sake of time, I'll just skip that. So looking a bit more at the case studies, why do we have this improved precision diversity trade-off? Well, I think the useful intuition is that MSA subsampling is creating this stochasticity by artificially injecting model uncertainty, right? The model is being withheld from having access to the full set of information. And so you would expect that maybe the model prediction will drift from the true ground truth structure. And in doing that drifting, it will sometimes drift one way and sometimes drift the other way. And that's what creates the diversity. But the overall prediction, the mean prediction from the MSA subsampling may drift. And that's, it's certainly not the case for all of these UniProt IDs, but at least for some of them, we do see some support for that phenomenon occurring. Whereas the AlphaFlow ensembles, they generally stay anchored around where the single structure prediction would have been, but just manifest some variance around that mean prediction. Now, I want to be clear that because the recall numbers are not really improving above the baseline AlphaFold, it is very rare that we actually see the ground truth conformational changes that are observed for these UniProt IDs actually recapitulated in the AlphaFlow ensemble. So I do want to be very upfront that this example that I'm showing here is very cherry-picked and certainly not the norm. Like normally, it's not quite this nice. But at least there are some examples of these conformational changes that are being accurately modeled. So here we have the PDB ensembles in black, labeled with these black triangles, and on the right-hand side, 4 randomly picked AlphaFlow structures. And we see that the general motion here is there's this rotation of this motif relative to the rest of the protein. We don't seem to capture the full extent of that rotation, but the general gist of it is there. Here we have, well, this is actually very hard to see, but there's these 2 helices which shift orientation a little bit relative to one another, but it's the helix that's behind the, there's 2 helices and it's the one in behind that's moving, so it's a little hard to see. This one here is maybe a bit more canonical. We have a transporter which is going through a conformational change, which at least from the top, it manifests as a little bit of a rotation of this inner barrel of helices. And yeah, once again, it's a little hard to see. We capture some of the motion, but as you can see from the PCA plot on the right, not quite the full extent. Now, for the second part, I want to take a look at ensembles from molecular dynamics. So this, in some sense, is maybe the slightly, at least in my view, more exciting part of the work, because for the PDB ensembles, perhaps one would say the MSA ablation methods are maybe not fully satisfactory, but clearly we're also not able to improve upon them that much, right? I mean, we improve upon them in the diversity, but in terms of capturing those observed conformational states in the PDB, we're also not quite there yet. The MD ensembles, on the other hand, though, represent a new kind of training data, which it's difficult to imagine that you would ever be able to replicate these ensembles well if you don't train on them explicitly, right? And previously, we didn't necessarily have even a framework for thinking about how to ingest MD ensembles and train them using something like an AlphaFold architecture so that we could try to recapitulate properties of those ensembles for unseen proteins. So that's what we're going to try to do now. To be clear, though, here, there are two related problem statements which we're going to evaluate with our method. So one is the problem statement where you don't have any template structure. And by template structure here, I would mean the structure that you use to initialize your molecular dynamics simulation, right? So in the absence of any single structure template, trying to recapitulate that output ensemble for molecular dynamics is the closest task to trying to model the Boltzmann distribution from scratch. And of course, this is a very hard problem. The slightly easier problem is the case where you actually supply to the neural network the model that you use to initialize the molecular dynamics simulation. And in this case, although you're still trying to capture a distribution of output structures, it's the distribution of output structures that are reachable within, for example, 100 nanoseconds of molecular dynamics simulations from that initial structure. So it's a little bit of an easier problem and closer to the use case that people actually currently have for molecular dynamics, where, of course, you need to supply this initial template structure. Right, yes, okay, I think I said these things. These evaluations are made possible by a very, very recent database that was published in Nucleic Acids Research just a few months ago. So, of course, as we all know, the PDB is essential to the success of AlphaFold and other methods. Previously, there really have been no well-established, publicly available data sets of molecular dynamics simulations for a large number of proteins. And so this Atlas data set is really what makes this work possible. And we anticipate certainly many other works in the future using this data set. But just to summarize what this data set is, we have explicit solvent all-atom MD simulations. So these were properly done simulations. These were not coarse-grained simulations or anything like that. And we have a healthy number of proteins chosen in such a way that they're maximally representative of the structural diversity in the PDB, with the exception of membrane proteins, which were not simulated. The simulations are carried out for 100 nanoseconds, so not trivial, but also certainly one wouldn't characterize them as long simulations. Yeah, so for purposes of the training and validation, we split them using the same release states cutoffs as we use for the main fine-tuning of AlphaFlow, just for consistency, right? So that we make sure that the test proteins are also things which our model has not even seen the PDB structure for. So, one question that arises is 300 nanoseconds long enough? And what exactly are we assessing? Is it meaningful? So, just to be very clear, 300 nanoseconds is certainly not enough to converge, have a fully converged simulation, right? We have 3 replicates, and for almost all the proteins, the 3 replicates actually differ quite a bit in some of their properties if you were to look at it in a very rigorous manner. So what we're going to assess is not how well we capture the equilibrium ensemble, but rather the equilibrium fluctuations around that single conformational state, which was used to initialize the simulation. And importantly, what we want to focus on is the ability to generalize to unseen proteins, right? Because this is eventually what we will want to do at the end of the day. And yeah, so we focus more on the generalization. We cannot assess, rare conformational transitions because these have just not been observed in the training set, right? So if it's not observed in the training set, there's no hope that we're going to be able to capture that at inference time. The training is just going to continue from the pre-trained weights that we obtained from fine-tuning AlphaFold on the PDB with our flow matching objective, right? So we take the alpha flow weights and then we just continue fine-tuning them for a little bit longer to obtain alpha flow MD. And this pre-training is actually necessary, right? If you were to take the original AlphaFold weights from DeepMind and immediately start fine-tuning on the Atlas dataset, the performance is quite a bit worse. So it's important for the model to first see conformational heterogeneity from the PDB to get its weights kind of primed for understanding what it means to take in this noisy input, and then we fine-tune the kind of stochasticity that's trying to model. Right, yeah, and again, just to be clear, the way that we've done the split is that the PDB IDs that are going to be used in inference time have not been seen at any stage of training, and they also represent a different ECOD domain from any ensemble that has been seen during training. Although there could be proteins with the same ECOD domain, which the model saw a single structure of in the first training stage. So there's a lot of models because there's a lot of training settings, but I'm just going to focus on the kind of the baseline evaluation that is most immediately implied by this training procedure. Right? Oh yeah, and for the runtime evaluations, we're going to look at the setting where we do have a PDB template supplied, because this is the setting where comparison with actually running molecular dynamics simulation is relevant. Yeah, so just to focus on the most important evaluations, we have three different kinds of evaluations that we want to look at when we ask how well do we emulate properties of these MD ensembles. And the first one of these is how well do we capture the gross flexibility of different parts of the protein? And we answer this by computing the correlation coefficients between the RMSFs observed in our sampled ensembles versus the ground truth ensemble, and comparing that to, for example, MSA subsampling. And here we see validation of the intuition that because MSA subsampling is injecting stochasticity by ablating the input, the regions of the protein that manifest as being flexible are going to be the ones where perhaps the coevolutionary signal is relatively weaker, right? It's not necessarily going to be regions of the protein that are actually more flexible when we heat the protein up to 300 Kelvin and simulate it, right? So we see that no level of MSA subsampling is actually able to meaningfully improve over AlphaFold on actually manifesting the residue level flexibility, whereas we have a pretty strong Pearson correlation coefficient as shown, and we just have a case study of a particular protein on the left here. Yeah, I'll skip this one. So this second evaluation is asking the question, how well do we model the gross motion, the principal axis of motion of the protein, right? And the way that this is computed is by taking the the 3 coordinates that describe the alpha carbon positions once you've done some RMSD alignment to the initial structure, and then we just do PCA over the over the motion in that 3-dimensional space. And we say, what is the cosine similarity between the PCA that we get from our ensemble and the principal component that we get from the ground truth ensemble? And this is just 1 minus the cumulative distribution of that distribution of cosine similarities. And the way to read this is that you just draw a vertical line and you say the value along this vertical line is the fraction of ensembles for which we recapitulate the principal axis of motion above a certain threshold. So again, here I want to highlight that AlphaFold itself already has some non-trivial performance on this metric, but ablating the MSA does not improve that performance, right? You get more randomness in your structures, but this randomness does not reflect in any way the principal axis of motion of that protein. And so we're able to substantially improve over MSA subsampling and the baseline AlphaFold in capturing that principal axis of motion. And although it certainly looks like we're pretty far from getting perfect recapitulation, I would also note that the cosine similarity that you get, or the success rate that you get from just comparing replicate simulations is also on the order of 50%. So this is actually pretty close to the best that you can do with these very short simulations. And finally, we want to make sure that the quantitative properties or the qualitative properties that we want to observe from these MD simulations are also recapitulated. So, for example, these rare events, quote unquote rare events, like these transient contacts or these weak contacts that we observe when we heat the protein up to 300 Kelvin, does that actually happen? And so here we just show that, well, actually this is, oh, right, okay. How is this slide organized? Okay, I'm sorry. I think this was the slide I was meant to discuss. Yeah, so, we want to see, for these proteins, in the middle column here, we see that in the molecular dynamics, we have these two residues, which are not normally in contact in the crystal structure. They come into contact for some non-trivial amount of time in the MD. In these two cases, we have some, interdomain motions. We want to see if these motions are also recapitulated in the AlphaFold ensembles. And the quantitative assessment here is that we are able to capture that to an extent that is better than MSA subsampling, although, still far from perfect. And you might also ask, the occupancy rates that we get from these ensembles, are they reflective of the underlying MD? In general, the answer to that is no. These two cases they align relatively well, but in general, it's only a quantitative or a qualitative statement that we can make, which is that, if the MD shows this happening more than 10% of the time, we will probably also show it happening more than 10% of the time, but we can't say anything more than that. For sake of time, I will skip the discussion with the runtime-based comparisons and to just show these evaluations, because I think that these evaluations also provide a slightly different way of assessing the quality of the matching the ensembles, right? Because the human eye is very good at just maybe picking up on that principal axis of motion and looking at the motions of various loops and helices and maybe transitions between different secondary structure types. Right, and you can just quickly glance at one ensemble versus the other and see if the property that you've kind of honed in on is captured in the AlphaFold ensemble. So this is the animation that's just in the GitHub, so maybe you've seen it already. But here is another one. This protein has a larger range of motion. We have these two domains kind of coming apart. And in AlphaFold, we are able to model that this is the principal axis of flexibility, although as you can see, the model still has a little bit of a hard time separating the domains as fully as they are in the MD. This here is another protein where there's some interdomain motion. We recapitulate the rigidity of the bottom domain and the flexibility of the top domain and the general flavor of the motion of these two domains. Maybe there's kind of a little, this little rigid latch here, which is maintained in its correct position. I think these plots or these animations, they give a very condensed idea of the level we're able to recapitulate these ensembles, which is I think an exciting progression of the state of the field, but certainly not perfect. Right, there are certainly some features that are not quite fully captured yet. Oh yeah, this is just another larger protein where we have the smaller domain on the left-hand side, which is kind of moving to the right for just one frame. And this we do actually capture pretty well. Okay, for sake of time, I'll just speed through this discussion and conclude. The basic idea of these MD evaluations is that we can produce ensembles whose fluctuations emulate those at the current timescale limitation of our training data. Hopefully, with more training data, we can do training and evaluation on longer simulations and recapitulate those longer timescale transitions. We, of course, cannot substitute for any application that requires explicit energies because we don't provide any kind of output which resembles an energy. This is just to clarify some misunderstandings that people have or misinterpretations of our claims. We also don't necessarily aim to sample the Boltzmann distribution per se, because we don't provide any information about the solvent, about any ligands bound, about any cofactors bound, the pH, the ionic strength, right? So many things are underspecified. We're not trying to recapitulate any particular Boltzmann distribution. We're trying to recapitulate plausible structures that the protein might take under some environment, right? Whether that's the crystallization environment or the particular environment that was sampled in the MD ensemble. For sake of time, I'll just end there and happy to take any questions or continue the discussion.
Well, thanks, Bowen. That was great. I feel a little bad that we didn't have more time scheduled for it. But I think you did a great job. Thank you. It's very impressive. I would like to open the floor for people to ask questions rather than me jump right in. There was one question in the chat. Let me see where that went. Jennifer Madagre, do you want to may I ask your question? A little technical question, but go ahead.
Yeah, just thinking about what you're measuring with the MD simulations. Did you do equilibration before the 100 nanosecond step, or is the 100 nanosecond step the part of the equilibration?
So these simulations were well, I didn't run the simulation, so I cannot answer this with 100% confidence, but my understanding is that they were equilibrated. They were solvated, brought up to a particular ionic strength, made, certainly neutralized, and then equilibrated. And the equilibration was the same for all three of those replicates. And so that's why we can say that there was like a common starting frame to those simulations. But yeah, my understanding is that those were equilibrated. Yes.
Okay, awesome.
Great. Feng Yu, go ahead.
Yeah, so I have actually two questions. So first question is that how many proteins have you tested with your model? Because even though you train it with PDB, MD, and minimize the RMSD, but it doesn't guarantee that your model will generate reasonable protein conformations from the biophysics perspective.
Yeah. So the evaluation is certainly not. It certainly could be evolved. It could involve a larger number of proteins. For the MD, we had 82. We had 82 test ensembles, right? So all of these statistics are averaged over those 82 proteins. And then I'm not going to be able to find the slide, so I'll just stick it at the bottom slide. And for the PDB evaluations, again, what we did was just take all the uniprot IDs, which had structural heterogeneity, and which were all deposited after that cutoff, right? Actually, the 2020 cutoff. So there's actually not a very large number of such proteins. So, although we use 100 uniprot IDs for the PDB ensemble, it would be great to use more, but I think we might just need to wait for a little bit longer until after the cutoff. The number will certainly increase, but that's the number right now.
Maybe, and to clarify my question, it's like I'm thinking about this whether each confirmation your model is generating is good, reasonable, or consider, or there's not, because you are using a noise, diffusion model is using a noise and denoise process. So there may be a lot of unreasonable confirmations, and in that case, it may impact the quality of generation. And another suggestion I have, let me. So another suggestion I have is that you mentioned that it won't be able to see the rare transition between confirmations because it never seen it in the training data set. But one thing is that in PDB, there might be some different confirmation for one protein. And more importantly is that in MD simulation, we're trying to model the flexible region. But in most PDB, it won't see the flexible region. So that's like we are trying to model the protein flexibility with some data that we will not be able to see. So that may be the most important part of modeling protein conformations. And so it doesn't mean sorry. So it doesn't mean that you cannot see the data. You should not be able to model it. That the question is that we cannot see the data, but we need to model the flexibility of flexible regions.
Point. Yeah. And I want to slip in a question here very related to that. So the original AlphaFold training excluded NMR structures. Now they're not a large portion of the database, but it's actually kind of nice that you can actually validate some methods against those NMR structures that were excluded. And I'm just curious, Bowen, did you exclude NMR structures from your training set as you continue to train AlphaFlow, or are they included also there?
Yes. So at the end of the day, they were excluded. This was not due to this was not an intentional decision. This was just I used the AlphaFold library to train, and just the I tried to make minimal modifications to their training setup, and so the filters that filter out NMR structures were that was kept. With that being said, as for the possibility of using the NMR structures as a held-out set, I think one challenge is that although the raw data for the NMR potentially could be very useful for the validation, my understanding is that generally for the NMR ensembles, there's a very small number of structures that are actually deposited, on the order of 10 to 20, and so that doesn't actually end up being enough to do the kinds of evaluations that I did for these MD ensembles. Right, because the flexibility seen in many of those NMR structures is also of the nature that you see from the MD, right? You have these maybe these flexible loop regions moving around, but if you only have 10 or 20 of them, that's just not enough to have any meaningful statistics. And so for that reason, I haven't really focused too much on using NMR structures for evaluation. It would be great if we could, the raw data could certainly help, but the actual confirmations are too limited.
A great point. And I just wanted to say, relative to what Feng Yu was saying, that if you have a crystal structure, if you have high B factors, you don't report those coordinates. So those flexible regions are not there. But with an NMR model, you report all the atoms that you have chemical shift data for. So even if they're very flexible, you'd still have atomic coordinates for them. But
yeah, okay, fair point. Yeah.
Let's move on. So there's a few hands up. I don't know who was first. Arnie, do you want to go ahead and ask your question?
Yeah, I mean, quite quick questions. I have two questions. First, I really like your precision recall curves, but of course, it's a bit disappointing that you couldn't do better. So what would you need to do better? And secondly, I just take the question, in your model at the end, do you do amber mineralization at the end? So or are there like risk for chemical bad parts of the proteins?
Sorry, I missed the second part of your question.
So the second part, like, I mean, in AlphaFold, normally you do an amber minimization at the end, basically, to avoid things clashing and bad stereochemistry. I couldn't find your paper if you do that or not.
Yeah, so my understanding is that it seems that as many people use AlphaFold, people seem to skip that minimization step or the relaxation step in Amber, and so we also did not do that. I don't anticipate that it would change any of these metrics, right? Because generally, as for it is disappointing that we're not able to do better. What would it take to do better? I think there are certainly a number of things that I'm exploring right now, and hopefully there will be some improvement soon. But it is a very hard problem.
Is it lack of data, or is it?
So there are a number of
one of the options, of course, is that you sample correct things, but that are not just seen in the database yet. But it's also, of course, you might do just you might also as well just add noise. It's like.
Yeah, that would certainly be a lovely possibility, but that's probably not the case. I mean, certainly having more data would always help, but I think there are some modeling choices that could do better even with the data that we have. And certainly this work is certainly not the end of the story as far as combining AlphaFold with generative modeling goes, and so we're hoping to be able to make some changes that improve the recall numbers.
So I'll call on Siva next, and then Alberto Perez. Go ahead, Siva.
Hi, thanks a lot, Bowen. That was a very great talk. So I was wondering, unfortunately, I didn't have time to read upon the data processing in terms of UniProt, but I assume that you just simply map each of the accessions of UniProt to individual PDB IDs, and I assume the length of the proteins is then constant, right? So it's just like you're comparing exactly the same domain, just in different conformations, or exactly the same entry in PDB. Is that correct?
Right. So that part is actually pretty tricky. So the mapping from the PDB seqres to the UniProt is from the CIFS database, and then I use the UniProt sequence as the reference for generating all the sample structures. And then to actually compute these plots, I do a multiple sequence alignment and keep the residues that show up in all the PDB chains and in the UniProt reference. And so, for example, if you have a UniProt ID where there's a very disordered region in the middle and it shows up in none of the PDB structures, that part will get cut out.
I see. Yeah, makes sense. And then the second part of the question, from your slide with PDB conformational ensembles. So I didn't really get, how do you exactly cluster like the models? Is it so you compute the precision like LDDT between prediction and the nearest PDB structure, but what is the metrics according to which you assess? Is it TM, or what exactly is the metrics?
So there's actually no clustering done for the evaluation. The clustering was just in the selection and the UniProt IDs. And frankly, maybe I shouldn't have done clustering there either, just to be consistent, but just more for historical reasons. The clustering was done to select the UniProt IDs, but then for the evaluation, there was no clustering.
But then how do you select the closest PDB structure? Is it template modeling score?
I'm sorry, the audio, I couldn't hear that.
Sorry.
Do I get what?
Can you hear me right now?
Yes.
So yeah, my question would be then, if there is no clustering, you have to calculate the precision for the nearest PDB structure, right? How do you find the nearest PDB structure? Is it a template score, or how do you compute it?
Oh, I see. Well, there's not that many. So this is actually, yeah, I compute all pairwise LDDTs. So, there's at most, 1,500 of them per UniProt ID.
Go ahead, Alberto.
All right, awesome talk, thank you. So I was wondering, we do a lot of simulations. We're involved with several databases for MD data. And I was wondering from your side, like, what would be your wish list of things that you would like? So you have like some short equilibrium simulations, but maybe there are other types of simulations. So if you could choose and you could ask someone to do the kind of simulations that would help you the most, what would you want?
Yeah, that's very interesting. I think one thing that is very important is just to have simulations for as much of the known protein structural universe as possible. So the simulations don't necessarily have to be super long. As you can see, there's already a lot of learning that we can do on short simulations, but just simulating more domains, that would be great. And also, I noticed that the Atlas data set here, these were just unbiased simulations, right? But I'm aware that there are, for example, AMD or Gaussian accelerated MD methods that maybe can give you more mileage in terms of getting more of the equilibrium ensemble for a certain number of simulated steps. And yeah, I was curious why the Atlas people didn't use some of those techniques.
Okay, awesome. Thank you. Maybe I'll connect with you later on.
Okay, that would be great. Thanks so much.
Thanks.
So, yeah, I just want to take just a few minutes before we're done to give an update on where we are with the CASP experiment. So, of course, which is really critical is being able to make predictions and evaluate them retrospectively, as Bowen described, but also prospectively. That is in a blind test. So in the CASP 16, what we've decided is we can't really evaluate these multiple conformation predictions against models because the models themselves had to be modeled from the data. So rather, we're aiming to assess against data. And it's challenging because we have to then be expert enough with the data, meaning we bring the data providers in as part of the assessment process. So right now we so far have 2 targets, and we're trying to get more. The first target comes from Bruce Donald and Terry Ois at Duke, and this is essentially it's a little bit different problem. It's 2 domains attached by a flexible linker. And the 2 domains hardly or maybe don't even touch each other. So really, it's really a modeling problem of the interdomain linker, although there's an excluded volume effect of the 2 domains. So they actually presented their approach and the kind of data they have in the last SIG that we had last month, and it's available online. And so in the YouTubes. And the idea is to assess against an NMR residual dipolar coupling experiment. The predictors don't need to know how that experiment works. They will be predicting ensembles, and we'll worry about how the assessment. We're also going to assess against SACS data that we expect to be able to collect together with John Tainer's lab. And there's one point that Bowen made that's really important here. These assessments, you not only have to get the different structures right, but if you assess against data, you have to get the populations right, the Boltzmann distribution. So it may be that one does a very good job of predicting alternative states, but the alternative state is not present in the data. It's only present when a ligand binds or only when it binds a partner. So that is a challenge in this assessment approach. We also have a second similar kind of a system. This is the BET protein has a domain called the ET domain, and it binds to a protein from a murine leukemia virus integrase. And this is just a picture of this idea of it. There are two domains connected by a flexible linker. There is NMR data on this, relaxation data. We're also going to collect SACS data. This picture is by no means a true representation of the conformational distribution. So those are two data sets that we have so far. But we really need to get more. And some people have indicated they may provide cryo-EM data particularly would be valuable here. There are some other NMR type experiments. X-ray crystallography, not so much, it could be multiple structures observed in different crystal forms or even flipping of side chains and maybe cross-link data. And I have to say, we're still in the process of getting more targets. Maybe some will come in through the natural CASP target collection. And maybe just before we end, I think Andre is still on. Andre, do you want to make some comments about this challenge about finding targets for this component of CASP? I know you may not have been ready for that. Maybe you even signed off already. I'm not sure.
Thank you for giving me that opportunity, guys. Sure, so we have a lot of people here, close to now 40 people. So we are in active search for targets for CASP15, and so targets, especially good targets, are getting rare these days. So we would encourage you to check whether you can find something in your circle. So it's always best to personally contact someone and ask for targets. So if you have colleagues who are working on some targets, on some interesting structures that can be used either for multi-conformational challenges in CASP or just a regular CASP target, interesting target, monomeric, multimeric, ligand, RNA, protein complex. So please ask them to donate those for CASP so that we can use them as CASP16 targets. And if any questions, so tell them to contact me at casp@predictioncenter.org, and I'll be happy to answer all the questions.
I want to make one more comment related to that. So typically in a CASP assessment, we have the coordinates and they're hidden in ground truth. And we don't want people obviously to publish those coordinates before the CASP assessment or make it publicly available. And that's the majority of targets that Andre is talking about. But for this project of multiple conformations, I don't think it matters so much that people have published their models of multiple conformations. If we have the data, we're assessing against the data. So a predictor may do a much better job predicting against the data. So I think we would be even willing to take structures that are where the model of the multiple conformation is even published, but we're going to be assessing against the data. We may find predictors do a better job, or CASP predictors, a better job than what's in your publication. So I just mentioned that as a thought I had recently. So this went great. Does anybody else, if anybody else wants to say a point, they could bring up, otherwise we'll stop here. We're up to the hour.
I just had a biology question. These systems you look at, are they interacting with other proteins in the cell, or are they really so flexible just?
These proteins do interact with other proteins in the cell, and in interacting, they probably their preferential relative orientations are important in the interaction, but probably they get frozen out when they interact with those partners.